Welcome to another episode of Tiger Bytes, where we talk to faculty experts from across campus about their areas of expertise and how they've used online learning tools to enhance the student experience. Today, we're with Lamar Siebenhener,
Very good.
who is a genetics expert. Is that fair to say?
Genetics, cell
Yeah.
biology, those are the two courses I teach.
Super. Thanks for being here.
Very welcome.
Um, just getting started here, could you tell us a little bit about what you've done in the online environment with regard to your teaching?
So, as far as online teaching, there's a lot of different uh, thoughts on how effective it is and everything. What we have been able to do, and our approach was somewhat that we were going to try to accommodate large enrollment classrooms and bring some of the ideas that have been used in traditional teaching of smaller classrooms, try to bring those into a large scale environment. Uh, that's where kind of the electrophoresis learning uh, process came in, the ELS system came in. It allowed us back again, ancient times when I was in school and everything, and we were doing labs for genetics and everything. We would actually run a gel, and we did that during the lab time, which is never an easy thing to do. It's expensive to do, and that was fairly small classes. That was 60, 80, 90 people. Now we're running class sizes of 250 to 300 and trying to have that wet lab aspect is somewhat difficult to achieve.
So, what would
In those
actually
things.
go on in one of these lab activities?
For just a wet lab,
Yeah,
the ideal way that we used to do it. Again, this was when I was in school and when I was actually in graduate school TAing the course. Uh, we would pre-make the gel for the lab, and then we would come in and put the gel. And when you run gels, you put them in a buffer system. So we would have a little cassette that we would put them in, a little box that we would put them in, and then each student would try to get up and they try to load the gel, which, as you can imagine, somebody who doesn't have any experience doing that doesn't always work well. But then we'd get the gel loaded, and it takes a period of time for the samples to move in this electrophoretic field. And then once they've moved, we would take it and I keep hitting the microphone here. We would take it and visualize it in some way, whether it was staining or something like that.
And
So
what's in the gel?
the gel is going to be some type of biological macromolecule, a protein, DNA, something like that. Mainly, we use DNA samples because kids always we would always try to isolate DNA. Kids like to look at DNA and everything because it really looks like a big ball of snot. Is how it was originally described. So that it's always kind of fun for them to look at that. But we would then run that DNA out on the gel, and if you treat that DNA in multiple ways, you can cut it up into smaller pieces, and you can compare this sample that was cut in one way with this sample that was cut in another way, so you can get different banding patterns of how much of what that DNA looks like.
And then what does that look like in the online environment?
So, an online environment, what we were able to do, Auburn Online did a really fantastic job with this and helped us do it. Uh, is it's kind of a point of view system. So they're looking at a computer screen where you can almost kind of see your hands in front of you.
Right.
So a typical point of view computer game almost, where you have to choose the samples that you want to load, and that's part of the learning process. You're given background information about a particular problem, and then we're asking you to take these samples and solve that problem. So you have to choose the samples, and you have to go through a preparation of the sample, which involves heating them up. So you have to put samples over a little Bunsen burner for a set period of time, And it's not just like you do it and let it sit and walk away. If you don't put it on there long enough, the results don't work. If you put it on there too long, if you heat them up too long, it doesn't work. You have to start over. So it somewhat simulates what we have in a real lab environment. Whereas if you mess your samples up, you have to start back over. So that's what they are going through. They have to prep the samples, then they have to load the gel, which is kind of a simulated pipette that they pick up. They load the samples into the gel, then they have to run the gel. Once again, if you run the gel too short, you don't get results. If you run the gel too long, you don't get results. And you have to go back to the very beginning and start back over. So I think it gives them a little bit of a feel, in a simulated environment, but it gives them a little bit of a feel where we can show them what it's like to kind of be in a laboratory setting, because you can mess up, you can make mistakes. And when you do, you have to, deal with that mistake. It's not like you just automatically you're going to get results every single time.
Right.
So it's a nice little system to allow them to visualize that, and then the results when they actually see the gel running, which is really kind of a cool thing to look at. You actually see your samples running in the gel, and then they get, they can print out images of what those samples look like, and then they can start trying to answer the questions.
So they're doing this on their own time at home?
We do this in a lab setting.
Okay.
So we have our genetics labs are 2 hours, 1 day a week.
Okay.
So they come in and they generally do it in that
On their
period
own
of time.
laptops?
Yeah, on their own laptops.
It's easy to see why that would be advantageous rather than having 250 actual lab stations
Exactly.
setting
So
up. You'd have to borrow the football field.
exactly. And even if we have generally 32 to 36 students in every given lab. So, you could only have 32 or 35 stations, or you combine them and say, okay, you 6 get to use this. So there's ways to cut it down, but still it's very time consuming to go through all that.
If you do the actual experiment, is that expensive? Like, is the gel costly?
It's much more costly than running it in a simulation.
Well, that's sort of what I was getting at.
But it's not as expensive as it used to be. If you're talking just simply running something like an agarose gel or something, which is very commonly used for these types of systems and everything, it's relatively cheap. But when you add the time that you have to put into preparing the gel, preparing the samples, getting everything set up, making the buffers, and doing all that, now you've got somebody's time involved in that, and then it becomes a lot more expensive.
So what were some of the most significant challenges you faced in translating the traditional hands-on lab approach to genetics into the online format?
Probably the most significant aspect to it was to make it look good, so that they really had the feel that hey, this is what a lab kind of looks like in it. So,
one of the things I was wondering was if you would have tried to do this 10 years ago, could it even have been possible, given how much the technology has advanced over the last decade?
I think it would have probably been possible 10 years ago, maybe 20 years ago. No, just because the coding, the writing, and again, I know next to nothing about doing
You don't
that.
need to.
But having something like Auburn Online that can provide that, is a really nice feature to have at Auburn University. But they have the expertise to give us something that looks like a laboratory setting and helps the students get a better feel for what's going on. So that's probably the most significant thing. It would be easy to just say, okay, kind of write up, this is what electrophoresis is, this is how you do it, here's results, go and interpret it. But I think it's just better for the student. I think they enjoy it more, they get a little more out of it if they can actually kind of almost feel like they're doing something a little bit.
Well, that leads me to the next question, which is, has there been any change in student performance since you went to this online approach?
I think so. I think the students have a better understanding of what some of these techniques are. Again, genetics is a 3,000 level course. So technically it's a junior level course, but
Hmm.
in all honesty, it's mostly freshman, sophomore. It's probably
Mm,
a very large sophomore population. A lot of these students have never been in a lab. They don't really understand how experiments work in a laboratory setting. So giving them an opportunity, as somewhat minimalistic as it is, because it is not a hands-on wet lab, but giving them the opportunity to visualize. These are the steps that you have to go to. We're going to make it a little bit easier for you to understand and a little bit easier to perform. But being able to visualize it, I think, really lets them um, gain a better feel and a better grasp for what you're actually accomplishing by doing electrophoresis. You may have people, listening that, okay, what is electrophoresis? If I can put you in front of a computer and I show you this is actually what's happening, and then give you the background information for you to read ahead of time to
right.
prepare. So you know what the DNA is, you know how you're treating the DNA, you know how you're getting your samples, and then actually see that run out on a gel. I think it's very beneficial for them.
Do you have a next generation version of this system in mind? Like what you'd like to do next?
Interestingly enough, we were just talking a minute ago when
Mm,
I came in. It would be very cool to uh, move this into, and certainly I think students would love it more, to move it into maybe a virtual reality type thing where
with
they
the
actually put the
goggles.
goggles
Yeah,
on, and then you actually have to
right.
go through
Yeah.
the mechanism of doing. I think that would be an exciting area to explore on doing this. That's what I would envision
Mm,
being the next generation.
right.
Kind of doing the same thing, but now you're not just clicking buttons with a mouse. You're actually, got and you're reaching over here and you're picking something up and having to manipulate it and everything. I think that would be a great approach to doing this.
It makes me think of the racing simulators that they have now, which are extremely
Oh,
realistic. But if you crash your car, there's no
yeah,
problem. You just
there's no
reboot.
real thing, but
That just shows you how realistic
it shows
the
you how
virtual
realistic. So,
reality.
so I do think if there would be a way to, write those types of programs to do this, I really think students would get a lot out
Oh
of
yeah,
that.
that's Auburn Online's challenge,
That's
not
Auburn
not
Online's
yours.
challenge.
In what ways, if any, has this project influenced your perspective on teaching STEM, particularly in terms of accessibility and innovation?
Yeah, it. I think it opens up some doors because now it's not just okay. I how can I teach a particular subject related to genetics, whether it's inheritance or whether it's molecular genetics, whatever. Now the thought process becomes, how can I apply that virtual reality world, which is kind of where we're going, uh, to doing that? How could you bring in like an AI system that could pose you on the spot questions, or something, I there are simulators out there for like nursing and everything where they can sit there and program in a problem
Hmm.
and you have to deal with it. I think that would be a cool thing to be able to do in a lab setting as well. Okay, okay, virtual reality, you run the system, but something goes wrong. All of a sudden, you get a spark over here. How do you do that? I
Right.
think that can make it more fun for them.
Hmm.
I don't know that that's necessarily driving home the points we always want to get across. But I do think making it more interactive is is a is more fun for the student. And I think if you can make it more fun and more interesting for them, that's where you start bringing in that uh, interest from the students, and having everyone having the ability to see those things.
I don't think there's any question that if people are more engaged
Right.
and
Engagement.
dare I use the word entertained,
Yeah,
they're going to learn more.
sure. And that I think that this offers the
Mhm.
possibility of that.
Have you received any unexpected feedback from students or colleagues?
Not so much from students. Every now and then, you're going to have that. You're teaching 250 students.
Law of large
You're
numbers.
going to have 10 students. I just don't get why we're doing this. This
Mhm.
is boring. Most of the students enjoy it. Most of the students get it and enjoy being able to see what's going on. Interestingly enough, from colleagues, I've almost had it be more of, hey, we really need to get back to the wet lab side of things, which is great. I love the idea of a wet lab. It's just really, really difficult to do with a large student population. And again, we typically, I'm teaching 2 sections of genetics this semester with close to 400
Wow.
students.
Yeah.
So, combined, actually a little over 400 students combined. So putting them in a wet lab, yes, that is a great way to learn, and they really get a lot out of that. Doing that 400 students,
Mhm.
one, where are you going to do that? Tell me on campus where
Right.
that's going to happen.
Right.
And it's just not, I don't think it's necessarily a feasible thing. I've had colleagues, we've got another module that we simulate. We use a fruit fly simulator. And again, it was a program that was written here at Auburn, where it simulates the mating of 2 fruit flies. Fruit flies is one of the major genetic models that we use to look at heritability, whether a trait is dominant or recessive, something like that. I can remember growing fruit flies, and by the time you open fruit flies and you have to look at them, because again, what a fruit fly is, very tiny, right?
Sure.
The hard is not just something you put in your hand, you look at, you have to put it under a microscope. Well, to get a fruit fly to stay still on a microscope, not always the easiest thing. So you have to anesthetize them. And there are different ways to do that. But one way is like put a little chloroform on the top. Well, you got 35 students in a room all trying to use chloroform.
Right.
You can see where this can run into problems. So we have we have that type of program that we can look at heritability with. That's been a positive. Although again, I've had colleagues say, "Well, I think they need to learn how to handle fruit flies." Now, I'm not sure that's what you need to be teaching them.
Because they're going to be handling fruit flies the rest of their lives.
Yeah, you get into a fruit fly lab, then you can learn how to handle. I want you to get the background information. I want you to understand the underlying basis of how that heritability occurs. And growing fruit flies doesn't do that. Analyzing the data you get does. So having ways that we can generate the data without having to go through all of the laboratory stuff, I think is a really important thing.
Lamar, thanks for being here.
You are very
Thanks for
welcome.
joining us on
I
Tiger
appreciate it.
Bites. We enjoyed learning a lot about biology and how it can be taught online, and talking about some other biological issues as well.
Yeah, I enjoyed it. Thank you so much for having me.
Thank you for joining us for this episode of Tiger Bites. We hope you enjoyed the conversation and gained some valuable insights. Please like, subscribe, and share this podcast with your friends and colleagues. You can find us on all major platforms, and keep learning, keep growing, and we'll see you next time on Tiger Bites.